For this question, the polarity or the prime numbers on the end of the sequences are very important. So I'm not going to write out the entire sequence that you provided me here, but I'll just do a little bit so you can get the idea of how this works. So the DNA sequence that we have is 5' on this end, you indicated. So I'm just going to write that down here. And it's 3' on this end. Now, it's important to remember that the DNA sequence is actually double-stranded, even though this is only showing you a single strand. So based on that, I'm going to go ahead and write the actual other strand of the DNA underneath this. And this one would have an opposite polarity, so we can write the opposite numbers on these ends. And then I'm just going to use the base pairing rules to fill this in here. And this would be my second DNA strand, the opposite strand, or what we call the complementary strand. Okay, so now when we need to make the actual RNA, the key here is that we need to make our RNA 5' to 3' always. That is the case. And the RNA needs to be made anti-parallel to the template. So what that means is that we have to copy it off of this one because that's the one that is anti-parallel. So I'm just going to write that here. So it has to be made with the one that's in the opposite orientation. So to do that, when I look at that sequence, notice that across from the T would go an A, across from the C would go a G, across from the A in this case is going to be a U because, again, with RNA, you're not going to have T's involved. And then if we just do the rest of the sequence, it's going to be AAC. So that would be the mRNA sequence that would be made based on this DNA strand. So again, the polarity, the 5' and 3', those are really important for you to be looking at when you have a DNA sequence and you need to copy it into an RNA sequence. And we also want to always remember, if we're only provided with one strand of the DNA molecule, there is really a second strand there, and we may need to also go through and actually write out what that second strand of the DNA is going to be like.